Hi everyone, my name is Veronica and I'm excited to present my research project, EEG as a non-invasive biomarker in a mouse model of Fragile X syndrome. So you've probably seen photos of this type of procedure before, where electrodes are stuck to a patient's head. This is called an electroencephalogram, or EEG, and they can be done on people of all ages, and they can also be translated to rodent applications. EEG uses electrodes to reveal neural activity in the form of waves, which can be divided into subgroups of frequencies corresponding to the level of neural activity. Its non-invasive nature makes EEG an attractive potential biomarker for neurodevelopmental diseases such as Fragile X syndrome. Fragile X is the leading cause of autism due to mutations in a single gene. Drug development in the field of neurodevelopmental disorders is commonly slowed by failures in clinical trials, although these trials are usually based on promising preclinical findings. Thus, we urgently need to develop effective translational biomarkers that will enable more informative clinical trials and translational success. So, what are biomarkers? They're indicators that capture the physiological changes which underlie the complex behavioral outcomes in patients. Biomarkers can help indicate if a drug is reaching its target and producing treatment responses. In other words, a biomarker is a useful tool for examining the efficacy of pharmaceutical interventions for neurodevelopmental disorders. EEG is a promising biomarker due to its consistent signal pattern in Fragile X patients and rodent models. Its non-invasive nature makes it appealing for clinical drug trials, as subjects would not require to undergo distressing surgical procedures. The extracellular signal-regulated kinase, or ERK pathway in the brain, plays a pivotal role in the development of autism and Fragile X. An inhibitor called PD can selectively target ERK, and pilot studies in our lab found that PD reversed behavioral deficits in mouse models of Fragile X and idiopathic autism. My study built on these findings and examined whether EEG signal can track improvements in a behavioral phenotype after PD treatment. My research aimed to examine if and what changes in EEG signal correlate to behavioral improvements. To address my objective, I used a cohort of 6 male Fragile X mouse models. They underwent simultaneous EEG and behavioral recordings, allowing me to correlate their behavior to EEG signals. I implanted each subject with an EEG recording device, and after recovery, they underwent behavioral testing, where the subject was injected with either PD drug or a vehicle dose and placed in a home cage for 50 minutes, which corresponds to the length of time needed for PD to take effect. A home cage was used in order to create a baseline for neural activity when the subject is in a comfortable environment. Then I placed the subject in an open field test arena for 10 minutes, as this test has consistently produced a strong behavioral phenotype in Fragile X mouse models. The subject's movement was tracked by a camera. After the test was done, I collected the EEG data. The treatment sequence was counterbalanced, and the final experimental procedure was the product of 4 months of surgical and experimental trial and error. Data analysis included splicing the last 5 minutes of EEG signals from trial 1 and 2. The difference in the power of the EEG signal of different frequency bands between PD and vehicle treatment was computed and correlated to the difference in behavioral parameters during open field tests between PD and vehicle treatment. I used a Pearson correlation test to correlate neural signals to behavioral parameters, and my results indicated that changes in EEG signal to the alpha and gamma bands correlated with changes in open field test behavior. So Table 1 represents the Pearson correlation coefficient between changes in EEG signal and changes in distance traveled in the open field test. The table shows different EEG frequency bands. Each frequency has a corresponding Pearson correlation coefficient, or an R value, which tells us how closely the EEG frequency correlates to behavior in an open field test. The closer that the R value is to 1, the more correlated the two groups are, and the p-value tells us how statistically significant the data is. If the value is less than 0.05, it is significant. For example, changes in the alpha band had a high correlation with rodent behavior, and this data was significant. In summary, I found a trend where changes in alpha and gamma bands were highly correlated to changes in behavior and were generally statistically significant. In other words, my results confirm that there is a correlation between changes in electrical signals and changes in animal behavior. In conclusion, my results using rodent Fragile X models demonstrated that EEG could be a promising biomarker for behavioral outcomes. By studying the reliability of a potential non-invasive biomarker for Fragile X, I hope to lay down the groundwork for future initiatives, including studying female cohort and sex-based differences. I hope my results would eventually translate to further testing on human patients with Fragile X and autism. I would like to thank the Alberta Children's Hospital Research Institute for supporting my project with a Summer Studentship Award, as well as the Faculty of Veterinary Medicine at the University of Calgary and the Hotchkiss Brain Institute.